All right, let's go.
Number 8. The escaped genes hypothesis. Viruses were once part of you. This is the oldest of the 3 classical theories, and in some ways the most intuitive. Every cell, bacterial, plant, animal, human, carries mobile genetic elements inside its genome, pieces of DNA that can copy themselves, jump to new locations within the genome, and occasionally transfer between organisms, transposons, plasmids, retrotransposons. These elements are selfish. They exist to replicate themselves. They do not benefit the cell. They just replicate. The escaped genes hypothesis, also called the progressive hypothesis or the escape hypothesis, proposes that some of these mobile genetic elements escaped from cells entirely. They acquired the ability to package themselves in a protein coat and move not just within a genome or between cells in the same organism, but between entirely different organisms. They became infectious. They became viruses. The evidence supporting this is real. Many viral genes closely resemble genes found in their host cells. Retroviruses, like HIV, use an enzyme called reverse transcriptase that is nearly identical to the enzyme used by retrotransposons in our own genome to copy themselves. The HIV genome and certain retrotransposons in human DNA look so similar that they appear to share a common ancestor. Horizontal gene transfer, the movement of genetic material outside of normal reproduction, is rampant in microbial life and may have been the mechanism by which escaped genetic elements accumulated the additional genes needed to build a functional virus. A 2025 paper from Oxford's Mathematical Ecology Research Group, analyzing the evolutionary dynamics of this hypothesis, concluded it is mathematically viable. Escaped genetic elements can acquire the properties of viruses through a plausible sequence of steps driven by natural selection alone. The weakness, it does not fully explain the origin of the capsid, the protein shell that surrounds viral genetic material. Capsid proteins have no clear equivalent among known mobile genetic elements. Where the first protein coat came from remains unresolved under this theory. Number 7. The regressive hypothesis. Viruses were once living cells. This theory runs in the opposite direction entirely. The regressive hypothesis, also called the reduction hypothesis or the degeneration hypothesis, proposes that viruses were once small free-living cells, bacteria-like organisms that at some point became completely dependent on larger host cells to survive. Over millions of obligate parasitism, they lost everything they did not need: their own metabolism, their own ribosomes, their own energy production machinery, and were reduced down to the minimum possible replicating unit, just genetic material and a protein coat. This is not an implausible process. It happens. Rickettsia are bacteria that have become so dependent on living inside host cells that they cannot survive outside them. Mycoplasma genitalium, the bacterium with the smallest known genome of any free-living organism, has a genome of just 580,000 base pairs. Obligate intracellular parasites consistently show genomic reduction over evolutionary time. The logic of regression is well supported by real examples. The discovery of mimivirus in 2003 dramatically strengthened this argument. Mimivirus was found infecting amoebae in a cooling tower in Bradford, England. Its genome contains 1.2 million base pairs and over 900 genes, rivaling the size of some bacterial genomes. Common viruses like influenza have around 13 genes. HIV has 9. Mimivirus has 900. It encodes enzymes for protein synthesis. It encodes DNA repair machinery. It encodes genes that bacteria have. Its typoisomerase genes are directly homologous to those found in soil bacteria, suggesting it evolved from a free-living bacterium that progressively lost non-essential genes while adapting to an obligate parasitic lifestyle inside amoebae. If mimivirus represents an intermediate stage in this regression, a cell partway towards becoming a virus, then the regressive hypothesis has a real living example to point to. Number 6. The virus first hypothesis. Viruses existed before cells did. This is the most radical of the classical theories, and it may be the most ancient truth. The virus first hypothesis proposes that viruses, or virus-like entities, existed before cellular life. In the RNA world, the period billions of years ago before DNA became the dominant genetic storage molecule, RNA molecules self-replicated themselves using the chemistry of the primordial environment. They competed, they evolved. Some accumulated, by chance, the ability to package themselves in protein coats. These protoviruses existed before any cell had formed. When cells eventually emerged, the protoviral RNA entities that were already present began interacting with them, infecting them, integrating into them, co-evolving with them. Viruses were not latecomers to life. They were there at the beginning. The strongest evidence for this comes from RNA viruses, viruses whose genomes are made of RNA rather than DNA. RNA viruses use an enzyme called RNA-dependent RNA polymerase to replicate. This enzyme has no clear equivalent in cellular life. No cell uses RNA to copy RNA in the way RNA viruses do. It appears to be an ancient innovation that predates the cellular machinery we know today. Its existence in RNA viruses, but not in cells, suggests it evolved independently of cellular life, possibly in the precellular RNA world before cells existed. The weakness of this hypothesis is that it requires complex RNA-based replicating entities to have existed before the chemistry that supports them: ribosomes, cell membranes, metabolic networks. Whether RNA replication could have been self-sustaining in the prebiotic environment without any cellular support remains actively debated. Number 5. The giant virus theory. The fourth domain of life. In 2003, researchers studying a bacterial-looking particle inside an amoeba in a Bradford cooling tower expected to find a bacterium. They found mimivirus, a virus so large it was initially mistaken for a small bacterium under a light microscope. Its genome of 1.2 million base pairs and over 900 genes shattered every existing definition of what a virus is. Then, in 2013, Jean-Michel Claverie and Chantal Abergel at Aix-Marseille University in France announced something even more extraordinary: 2 new giant viruses, Pandoravirus salinus, found in sediment at the mouth of Chile's Tuncan River, and Pandoravirus dulcix, found in a freshwater pond near Melbourne, Australia, with genomes between 1.9 and 2.5 million base pairs and up to 2,500 genes. Pandoraviruses are visible under a standard light microscope. They are larger than some human cells. Their genomes are larger than the genomes of many living organisms. The most extraordinary detail: 93% of Pandoravirus genes are not found in any other organism on Earth. Not in bacteria, not in archaea, not in any known cellular life. They are completely novel genetic sequences with no known relatives anywhere in the tree of life. This led a group of researchers, including Didier Raoult at URMIT in Marseille, to propose that giant viruses represent a fourth domain of life, distinct from bacteria, archaea, and eukaryotes, an ancient lineage of life so different from everything else that it cannot be placed within the existing 3-domain framework. They proposed a new taxonomic order, Megavirales, to contain these organisms. Most of the virology community has not fully accepted this classification. Giant viruses still cannot replicate without a host. They still lack ribosomes. They still fail the traditional criteria for life in ways that bacteria do not. But the discovery of Pandoravirus confirmed one thing beyond dispute: the viral world is far larger, far ancient, and far more diverse than any model before 2003 had imagined. Claverie himself said, "Finding a new type of virus this different happens once every 50 years. Number 4. The Viroid Theory. The simplest replicating thing that has ever existed. A viroid is not a virus. It is smaller than a virus, far simpler. A viroid is a single strand of circular RNA with no protein coat at all, just naked RNA. between 246 and 401 nucleotides long, the smallest known self-replicating entities in biology. They infect plants. They cause diseases, including potato spindle tuber disease and coconut kidang disease, which has killed tens of millions of coconut palms across the Philippines. The RNA of a viroid has no protein-coding genes. It does not code for any protein. It replicates purely through its RNA structure. Its secondary folding causes host enzymes to copy it without the viroid needing to provide any protein machinery of its own. The Viroid Theory proposes that viroids are evolutionary relics, remnants from the RNA world that predate both cells and viruses. On this model, the sequence of events was viroids first, then protoviruses that acquired protein coats surrounding viroid-like RNA, then full viruses, then cells. The discovery of a viroid-like satellite RNA, HDV, the hepatitis delta virus, that infects humans using the machinery of hepatitis B virus gives this theory a living example to examine. HDV has a circular RNA genome of only 1,700 nucleotides. It is the smallest known pathogen affecting humans. It replicates using a mechanism directly comparable to plant viroids, a viroid-like entity infecting a mammal, living evidence that this RNA architecture is not limited to plants or to ancient history. Number 3. The endogenous retrovirus theory. 8% of you is ancient virus. This is the one that changes how you think about your own body. 8% of the human genome is made of viral sequences, not contamination, not infection, permanently integrated ancient viral DNA that has been in our genome for tens of millions of years. These are human endogenous retroviruses, HERVs. They arrived when ancient retroviruses infected the germ cells of our ancestors, the eggs and sperm, and integrated their DNA into the host genome. That integration was then inherited by every subsequent generation. The virus is now part of us, permanently. There are over 100,000 HERV sequences in the human genome. Most are silent. Accumulated mutations have disabled their ability to produce functional proteins, but not all. Synctin-1 and Synctin-2 are proteins encoded by HERV sequences, ancient viral envelope proteins that human biology has repurposed to build the placenta. The syncytiotrophoblast, the outermost layer of the human placenta that interfaces with maternal blood, forms through the fusion of individual cells into a multinucleated sheet. Synctin-1 drives that fusion. Without it, placental development fails. Without the placenta, mammalian pregnancy as we know it does not exist. The protein that makes you possible is a repurposed viral gene from an infection that occurred over 25 million years ago. The endogenous retrovirus theory of viral origins argues that this integration process works bidirectionally. Host genome can escape back into circulation as new viral particles. Ancient HERV sequences buried in our genome may occasionally produce functional particles that escape cells and infect other organisms, generating new viruses from host genetic material and a reversal of the escaped genes hypothesis. Viruses and genomes are not separate things. They are in continuous exchange.
Number 2. The coevolution hypothesis. Neither came first. Both did. All 6 theories presented so far assume some version of sequential origin. Viruses evolved from cells, or cells evolved from virus-like entities, or viruses evolved from genetic material that escaped from cells. All of them imply an order of events. The coevolution hypothesis rejects this framing entirely. It proposes that viruses and cells did not evolve one of the other. They evolved simultaneously, together from the same primordial chemical environment. The early Earth, before any life existed, was a complex chemical soup of nucleotides, amino acids, lipids, and mineral surfaces. RNA molecules began replicating. Some clustered with protein molecules to form primitive cells. Others replicated without assembling into cells, as free genetic elements. Both lineages developed simultaneously.
neither is ancestral to the other. In this view, viruses are not degenerate cells or escaped genes or ancient precellular relics. They are a parallel form of life that co-evolved with cellular life from the same primordial conditions and has been co-evolving with it ever since. The evidence is indirect but real. Structural analyses of viral capsid proteins, the protein shells that surround viral genetic material, show that the same capsid architecture appears in viruses infecting organisms from all three domains of cellular life. Bacteria, archaea, and eukaryotes are infected by viruses whose capsids share the same geometric fold. If viruses had evolved separately in each domain, they would not share this structure. The conservation of capsid geometry across billions of years and across all domains of cellular life suggests viral lineages are genuinely ancient as cellular life itself.
Number 1, the multiple origins theory. All of the above. This is not a cop-out. This is the position held by the majority of virologists working today, including the authors of the most comprehensive 2025 review of viral origins published in the journal Pathogens. There is no single origin of viruses. RNA viruses, DNA viruses, and retroviruses are so fundamentally different from each other in their genome structure, their replication mechanisms, their protein machinery, that they almost certainly did not arise from a single common ancestor. The virosphere, the total diversity of all viruses on Earth, shows no single universally conserved gene, no protein that every virus shares. This is in stark contrast to cellular life, where ribosomal rRNA sequences shared by all bacteria, all archaea, and all eukaryotes confirm a single common ancestor for all cellular life. The absence of any universal viral gene means the most likely explanation is independent origins. RNA viruses may have originated in the pre-cellular RNA world. Large DNA viruses, like mimivirus and pandoravirus, may have originated through aggressive reduction of ancient cellular organisms. Retroviruses may have originated as escaped mobile genetic elements that acquired protein coats. Different families of viruses evolved through different processes at different times. This makes viruses unique in biology. Every other category of living thing traces back to one common ancestor. Viruses may have no single ancestral origin at all. They are a functional category, things that replicate inside cells using cellular machinery, not a genealogical one. Multiple different lineages arriving independently at the same parasitic lifestyle, the same solution discovered many times by many different routes across billions of years of evolution. 8 theories: genetic elements that escaped cells and became infectious, living cells that regressed into pure parasites, ancient RNA entities that predated cells entirely, a possible fourth domain of life with 2,500 genes and no known relatives, naked RNA molecules that may be the simplest replicating thing that has ever existed, ancient viral DNA permanently integrated into your genome that builds your placenta, a parallel lineage that co-evolved with cellular life from the very beginning. And the most likely answer, that there is no single answer because viruses have no single origin. 8% of your genome is ancient virus. The protein that lets you exist as a mammal was coded by viral DNA from an infection 25 million years ago. Viruses have been here at least as long as cells have, maybe longer. Where they came from, we are still working on it. Subscribe to Cosmic Hives. See you in the next one.